Hi everyone, hope you're well. Welcome to the sixth lecture of MD 6028. This lecture kind of marks the start of the second portion of the module. So in the first portion of the module, or the first segment of the module, we were discussing topics that are central to virology, so fundamental, foundational topics that we need to understand before we go further into the module. So these are just topics relating to what viruses are, how we distinguish between the different viruses. And we actually looked at these topics in quite a bit of detail. We really went into some depth when understanding these distinctions. So my hope is that at this point, we really understand these key ideas within virology. We understand that there are different groups of viruses. We understand the differences between the groups in a significant amount of depth and in a really intelligent way. Now in the second portion of the module, I want us to take that knowledge and apply it to human disease. So this second portion of the module considers viruses as causative agents of human disease. So what we're going to do is just explore concepts relating to the ability of viruses to cause human disease. So I'm sure we know that viruses are pathogens. Many viruses are capable of causing human disease. What we need to do is understand how they cause human disease and then further understand what we can do about it. So we're going to start off in this lecture discussing again some of the key topics that we need to approach at the start when we're considering viruses as causative agents of human disease. We're going to consider things like virulence and pathogenesis. So these key ideas that we need to understand straight away. But then we're going to move on and we're going to look at infectious disease caused by viruses in a more rounded way. So we're going to think more about things like diagnosis, and we're going to think about the different treatments and the different therapies that can be used, as well as epidemiological approaches like public health measures that can control the spread of viral infections. So yeah, that's what this portion of the module is going to focus on. And again, today we're going to be focusing specifically on establishing some of these key definitions. Okay, so you can see some of these definitions defined here, things like pathogenesis and virulence. You probably have some idea of what these things mean, but we need to really establish them at a level 6 level. So we really need to understand them in a significant amount of detail. I suspect that if I asked you the difference between virulence and pathogenesis, you might know the answer, which would be great, but I suspect a number of students wouldn't, because it's in some ways it can feel like a technical definition, but when we're at level 6 and when we're going into a significant amount of depth, these definitions become really important, and so that's the kind of thing we're going to talk about today. Okay, so first off, viral pathogenesis. Pathogenesis means the ability to cause disease and the way disease is caused. So if we're thinking about the pathogenesis of a particular virus, we're thinking about how that particular virus causes disease. Pathos meaning suffering, it tends to mean a disease, de facto disease when we're using it as a medical term, pathos, disease. Genesis means the origin of something. So again, oncogenesis, the origin of cancer, with onco meaning cancer. Oncologist is a doctor who studies cancer or someone who studies cancer. Tumorigenesis, the origin of tumors. Pathos, however, as we said, pathology, the study of disease. So when we're saying pathogenesis, we're thinking about the origin of disease. And if we're thinking about the pathogenesis of viral infection or viral pathogenesis, the pathogenesis of viruses, we're thinking about how specifically viruses cause disease. So how does a virus, when it enters a host, result in disease? Viral pathogenesis. It's quite a complex question to answer, and it kind of makes sense that it's a complex question to answer, because if we think about viral disease, it becomes immediately obvious that we're talking about a wide spectrum of disease. So when we're fixing on a definition for viral pathogenesis, when we're explaining viral pathogenesis, we need an explanation and an understanding that is sufficient to explain how poliovirus causes disease, and rotavirus, and rabies virus, and HIV. We need a definition that fits all of these different viral infections. And that can seem challenging on the surface because these viral diseases are so different. The symptoms of these viral infections are so different. But what we actually see is when we look into them in detail, the same principles are playing out over and over. In a similar way to when we were looking at things like genome expression and replication, we were seeing different viruses complete genome expression and genome replication in different ways. However, they're sticking to the same principles. The same principles apply because fundamentally they're all viruses. The same is true for viral pathogenesis. We see the specific diseases caused by the different viruses are different. However, we see the fundamental principles are the same. What I have on the side here is a distinction between curative treatments and symptomatic treatments. It's an important distinction to make because it reinforces the need to understand how and why different viruses cause disease. There can be a kind of temptation to think we don't need to know the specific details of how viruses cause disease. We don't need to know that this viral protein does this and this viral protein does this. And I kind of understand that because from one perspective, if we're trying to cure infection, all we want to do is get rid of the virus. It doesn't matter how it's causing disease, really, right? All we want to do is inhibit the virus from replicating. So if we can get drugs that inhibit it from replicating, then that's job done, really. We don't need to worry about the symptoms and the other things the virus is doing because we're attacking it at the source, we're inhibiting replication. However, for a lot of viruses, we can't do that. We can't cure a lot of infections. Also, just because we're curing an infection, we potentially have to deal with the symptoms as well. A cure isn't necessarily immediate. So we tend to have a focus on symptomatic treatments as well. So these are therapies that aren't necessarily targeting the virus directly. They're not inhibiting the virus's replication. So they're not working like antivirals, so antiviral drugs. What they are doing is treating the symptoms. So they are treating the patient, they are helping the patient, but they're not curative. So we just need to distinguish between curative treatments and symptomatic treatments. When we're talking about symptomatic treatments, it will become clear that they're not curative, if that makes sense. When we're talking about curative treatments, we'll be generally talking about antivirals. We'll be talking about things that specifically target the viral replication cycle. When we're talking about symptomatic treatments, we're not generally targeting the replication cycle. We're talking about other processes within the body that the virus is impacting. So again, back to why we need to understand viral pathogenesis. One of the key reasons is that if we want to treat the symptoms of infection, we need to understand what those symptoms are. And we also need to understand how and why the virus is causing those symptoms, right? All this information is useful, it's beneficial, it aids our attempts to develop and utilize therapeutic interventions that target symptoms of infection. Okay, so we'll talk about pathogenesis in a little bit more detail here. We'll just try to explain the concept of pathogenesis. I alluded to the fact that we have lots and lots of different viruses that infect humans, and these viruses can cause a wide variety of human diseases. And I rushed through a few different examples like poliovirus and rotavirus and rhinovirus. And these are viruses that cause very, very different symptoms. So they therefore have very, very different pathogenesis. They cause disease in a different way. Why is another question. So why does this happen? Why does this virus cause this disease? And why does this virus cause this disease? The answer to that question is quite varied actually. So there's a number of different factors that impact what type of disease a virus causes. And I've listed some of these factors here. And once we talk through them, it'll become, pretty clear why these factors impact how a virus causes disease. I have this life cycle on the right here, which we've talked about a few times. And we've said this is a generic life cycle, so viruses engage in this life cycle. All viruses engage in this life cycle. However, what I'm saying now is, despite the fact that all viruses engage in this life cycle, viruses cause different disease, so different viruses cause different disease. Again, how is this the case if they're engaging in the same life cycle? And the answer is because they engage in this life cycle in a slightly different way. And a lot of these factors I've listed on the side of the screen here, they are related to the life cycle. They are related to how the virus engages in the life cycle. This first option here, or not option, this first statement here, tropism with a question mark. The tropism of a particular virus strongly impacts what type of disease it causes. And what we mean by tropism is what cell type is the virus infecting. So all viruses engage in this life cycle, fine, but what cell type do they engage in this life cycle with? It's different. Different viruses will infect different cell types. Different viruses will complete this life cycle in different cells. Poliovirus, again, an example we've used, completes this life cycle in neurons. One potential symptom of poliovirus infection is paralysis. That's linked to the fact that poliovirus is tropic for neuronal cells, and tropism is, again, it's a term used to describe the cell type that a virus infects. So poliovirus is tropic for neuronal cells. Poliovirus has a tropism for neuronal cells. Either way we say that, it's fine. We're saying the same thing really. We're saying poliovirus is able to infect these neuronal cells, specifically motor neurons. If you think about other viral examples, what do we have? Hepatitis B virus. Hepatitis B virus is tropic for liver cells, for hepatocytes. Hepatitis B virus, therefore, results in damage to the liver. It results in liver scarring. It results in cirrhosis and hepatitis. These are symptoms of the liver, and the ability of the virus to cause these symptoms is linked to its tropism. It's linked to its specialization to infect hepatocytes, liver cells. Lytic versus non-lytic is the next statement here. And this is an interesting one, inherently linked to the life cycle, because some viruses, when they engage in this life cycle, they will lyse the cell. They're called lytic viruses. So by completing this life cycle, they will cause the cell to rupture and die. Some viruses won't. Now, whether or not viruses have a lytic or non-lytic life cycle impacts the type of disease they cause, and that, hopefully that's kind of almost self-explanatory. If a virus is infecting cells and lysing those cells, so it's killing those cells, that can cause disease in a very specific way, because it's essentially depleting the body of a particular cell type. It's causing localized damage. So another interesting point here is the immune response. So we need to understand that a lot of viruses, they don't directly cause disease. They indirectly cause disease via the action of the immune system. So the immune system reacts to the presence of the virus, and it's the immune system's reaction that causes the disease. A great example of this is hepatitis B virus, which you kind of just touched on. As we said, hepatitis B virus infects hepatocytes, it infects liver cells. It doesn't directly cause disease at this site, however. It's a non-lytic virus, so it doesn't destroy the liver cells. The immune system, however, detects the presence of the virus in these liver cells, and it attempts to clear infection, so it reacts to the presence of the virus, and in doing so it causes significant damage to the local environment, and this damage to the local environment is experienced as cirrhosis, so as liver scarring. So it's quite clear from that example how hepatitis B virus isn't directly causing the damage. However, it is responsible for the damage just via the immune system's response. So that's why we say it's indirectly causing the damage. So we also have chronic versus acute, and then this idea of latency. So we need to consider that not all viruses cause disease straight away. Some are able to persist, so we have a long-term infection that could potentially be characterized by reactivations. So if we think about HIV, HIV is a latent infection. It can go into this latent quiet state for like a decade. Herpes simplex virus also has this latent stage. However, herpes simplex virus is characterized by potential reactivation. So it's in this quiet state, but it can reactivate and kind of burst into a symptomatic state occasionally. And these symptomatic states are just breaks in this long latent stage, really. We also have to consider viral variants as well. So when we're considering a particular virus, we have to remember that not all individual virus particles that belong to a particular species are identical. We also have things like strains and subspecies and quasi-species, and it gets quite complicated, but this is a factor that can impact pathogenesis. And so we'll have variants of a particular virus that are more or less capable of causing disease. So I've got made a point here, and I'm starting to distinguish between pathogenicity and virulence. So a lot of these factors can impact not just pathogenesis, but also virulence as well. So we'll establish this definition now. Pathogenesis is the way a virus causes disease. Hopefully that's clear from what I've said. It's the type of disease the virus causes. So if you have a particular virus that infects a particular cell type and it causes those cells to lyse, and then you get scarring at that site, and then you got symptoms linked to the scarring. What we've just explained is the pathogenesis of the virus. We've talked about how it causes disease, and we've talked about what the disease is. That's pathogenesis. Virulence is slightly different because we're talking about the degree of disease. We're talking about the severity of disease. So if we're talking about the virulence of a particular virus, we're talking about the degree of disease it causes. That's distinct from the pathogenesis. So we can also think of it as the virulence is the degree of disease, the pathogenesis is the type of disease. So what I want to do now is look at some different viruses known to cause disease and kind of explore how the factors we looked at in the previous slides do actually impact pathogenesis. So we're going to look at the types of disease these viruses cause and explore which factors impact their pathogenesis. The example here we have norovirus. So norovirus is a cliquey virus. It belongs to the Cliquiviridae family. We can see it mapped out in the bottom right. It's in the RNA virus family, and it's a positive sense single-stranded RNA virus that is unenveloped, so it doesn't have a lipid envelope. So norovirus infects the intestinal brush border, so it's infecting the border of the intestines. It's pretty clear to see how its ability to infect the border of the intestine is linked to its symptoms, so linked to the pathogenesis, because it causes severe gastrointestinal symptoms. So it's transmitted through the fecal oral route, so it's ingested. And then when it's ingested, it infects the cells essentially lining the intestine. And once it infects these cells in the intestine, again, the intestine is a core structure within the gastrointestinal tract. it causes gastrointestinal symptoms, diarrhea, vomiting, cramps, nausea. These are all gastrointestinal symptoms in this case. We've said the type of disease that a virus causes, so the pathogenesis of a virus is linked to the cell type it infects. This is an example of that here. We've also said it can be linked to whether or not a virus is lytic or non-lytic. These noroviruses are lytic, so what they're doing when they're infecting these cells in the border of the intestine is they're disrupting them to the point where they are lysing them, so they are completely destroying them. This destruction results in malabsorption, so there's no absorption of water or nutrients from the food that's kind of passing through the intestine, and ultimately this is experienced as diarrhea and vomiting and cramps, as we said. So quite a clear example of how we can tie the pathogenesis of a virus to some of the factors we looked at in the previous slide. So the cell type it infects and the type of infectious cycle it has, whether it's lytic or non-lytic. Here we have hepatitis B virus, which we've talked about already this lecture, so I don't really want to go into any more detail here. I just think it's useful for you to have the information on the slides. Next up we have influenza, which is one of the orthomyxoviruses, so belongs to the family Orthomyxoviridae. RNA virus. It infects respiratory cells, specifically epithelial cells that are lining the respiratory tract. You know, we all kind of know the symptoms of influenza, and it's pretty clear that they're linked to the site of infection. So influenza virus infects epithelial cells of the respiratory tract. Then there's a localized immune response. This immune response is at least partially responsible for the symptoms experienced, you know, sore throat, cough, a host of other symptoms that are respiratory symptoms that are caused at least partially by the immune response. We also have to consider that the infection is lytic, so the virus does cause lysis of these cells. However, that's not the primary cause of the symptoms. The primary cause of the symptoms is the immune response. If we understand what inflammation is, we'll understand that a sore throat and kind of a lot of localized heat and pain is a symptom of inflammation. That's what localized inflammation is: redness, swelling, pain, warmth, edema. And we'll talk about these things in a bit more detail in later lectures when we look at the immune response. But for now, we just want to understand that a lot of these localized symptoms in this case are due to the immune response, part of which is inflammation. Next up, we have human immunodeficiency virus, HIV. HIV is another clear example of how we can link the earlier factors we discussed, which are largely dependent upon the life cycle of the particular virus, to the pathogenesis of the virus, so to the type of disease that the virus causes. So HIV specifically infects T cells. Primarily, we're talking about CD4 T cells of the immune system. So these are immune cells that have the correct receptor, CD4, that HIV can infect. When HIV infects these cells, it is at least partially a lytic infection. So the viral infection does cause some lysis. However, it also causes widespread changes in the immune system that ultimately result in a greater level of lysis, so a greater loss of immune cells. But ultimately, the end result is a depletion of these immune cells. So by infecting these CD4 T cells, HIV is mediating the eventual loss of immune cells. This depletion is it's characterized by an increased susceptibility to additional infections. So if an individual doesn't have enough immune cells to have a functioning immune system, they are susceptible to additional infections because they are immunodeficient. They have acquired immunodeficiency syndrome. which is characterized as AIDS. So again, pretty clear here how we can link the ability of HIV to infect immune cells specifically to its ability to induce immunodeficiency, aka its pathogenesis. Next, we have rabies virus. So rabies virus, it belongs to the Rhabdoviridae family. It's transmitted by saliva, so individuals generally contract rabies when they're bitten by a rabid animal. So when they're bitten by an animal that's carrying the rabies virus, the bite pierces the skin, and then the virus passes through the skin in the saliva. What happens then is the virus actually migrates up through the neurons until it reaches either the brain or the spinal cord. The reason this is linked to pathogenesis is you primarily see neurological symptoms in rabies. So the virus causes neurological symptoms, things like anxiety, hallucinations, hydrophobia, so this fear of water, aversion to water. So it's originally rabies was just termed hydrophobia. It was just called hydrophobia because it was the most obvious symptoms. You get individuals infected with this virus who they can't even be exposed to water without being overcome with anxiety and fear and panic and things like that. So this really severe aversion to water, which is very unusual, obviously, and very characteristic of rabies virus infection. And again, these symptoms, because we also have things like anxiety and hallucinations, primarily neurological, and again that's because rabies virus is infecting the nerve cells and it's ultimately infecting the brain and the spinal cord, and obviously rabies is ultimately fatal. That's the ultimate pathogenesis of rabies virus. So you'll notice that whilst hopefully you're convinced these factors are all kind of important in pathogenesis, they impact pathogenesis to a degree. One factor we've seen crop up over and over again is this idea of tropism. So this idea that the cell type that a virus infects is particularly important. It's a particular impactful determinant of pathogenesis. So what I want to do now is just zoom in a little bit more on this idea of tropism and just understand what exactly determines which cell type a virus is able to infect. So to do this, we want to consider the early steps of the viral replication cycle. We want to consider the steps of attachment and entry. These steps, as demonstrated here, are the steps that are pertinent to the ability of the virus to enter into the cell and which cell type of virus is able to enter. So these are the steps where the virus attaches to the outside surface of a particular cell and then enters into the particular cell. A term we need to understand here is susceptibility. So susceptibility is this idea of which cells the virus can attach to. So we're just talking about this initial process of attachment. So this initial process of attachment determines whether or not the virus can get into the cell, because it's the first step. And this process is itself determined by the presence of receptors on the host cell surface. So if the correct receptors are present on the host cell, then the virus can attach. If they're not present, then it can't. So this is the first step that determines which cell types a virus can infect. So if we have an unsusceptible cell, so we have a cell type that doesn't have the correct receptors, then the virus can't attach. If we have a susceptible cell type, then the virus can attach. And again, this is the first stage of the infectious cycle. So it's the first stage going ahead. So it opens up the potential that the rest of replication can occur. So the potential that the virus can complete the rest of its replication cycle. in this cell type. We need to think about co-receptors as well. So when we're talking about the virus attaching to a single receptor, we're oversimplifying things a little bit, just because most viruses will also attach to co-receptors. So they will only attach to a cell that has the correct receptors and co-receptors. So attachment is a little bit of a more complex process than a virus attaching to a single receptor. We just have to bear in mind that there's also co-receptors involved. Next, we have this concept of permissivity. Now, permissivity is linked to susceptibility, because permissivity and susceptibility both impact tropism. So a cell type must be susceptible to infection by a particular virus, and as we just discussed, that means the virus can attach to the outside of the cell and then presumably enter. But the cell type must also be permissive to infection, and this means, can the cell type sustain the virus's replication cycle in the intracellular environment? So we have a cell type that is susceptible to infection, so the virus can attach and then presumably enter into the cell. But can the virus then do all the other things? Can it express its genome? Can it replicate its genome? Can then it assemble new virus particles? All these are elements of permissivity. And to establish tropism, so to have tropism, we have to have susceptibility and permissivity. It's not enough to just have susceptibility and not enough to just have permissivity. There needs to be both. If we have a virus that can attach to the surface of the cell and presumably enter into the cell, so we have a cell type that is susceptible, but the cell type isn't permissive, so the virus can't replicate within the cell type, then we don't have tropism. Then the virus can't successfully infect that cell type. Hopefully that's clear. So when we're thinking about tropism, we're thinking about a combination of permissivity and also susceptibility. And for a very clear example of how a cell type can be susceptible and permissive, we have the epithelial cells of the respiratory tract, and we have influenza virus. So we know influenza virus infects epithelial cells of the respiratory tract because we talked about this earlier. So these cells must therefore be susceptible and also permissive to influenza virus infection. The susceptibility part is fairly simple. They just have the right receptors. So influenza virus is able to attach to the appropriate receptors and then it can enter into the cell. The permissivity is a little bit more complex, but hopefully it'll be really clear how this permissivity is present. So the epithelial cells in the respiratory tract are surrounded by a protease called protease clara that's being secreted by nearby cells. If influenza virus were to infect a different cell type that had the same receptors, so cell types that were susceptible but were not in this physical location, it would find this cell type is not permissive because the protease clara that is in the environment actually processes the new virus particles that are released at the end of the infectious cycle. So this protease clara processes these newly released influenza virus particles in a way that makes them infectious. If we take protease clara out the situations, there's no protease clara there. So in a different environment, we're talking about cells that are in a different part of the body. Infectious influenza particles won't be produced because they haven't been processed by protease clara. So in this environment, even though the cells are susceptible, so even though the influenza virus can attach to the cells and enter them, these cells aren't permissive because right at the end of the replication cycle, just as they're being released from the cells, so just as they're at the very last stage of the replication cycle, they hit a barrier because they're not being processed by protease clara, so they are not turning into infectious particles. So we can't consider the cell type permissive in this environment. So even though this process isn't quite in the intracellular environment, it's happening just after release, it still counts because it's still a stage in the replication cycle. We're having non-infectious particles being released, so therefore the full replication cycle isn't complete because the particles are non-infectious. So that cell type we would consider non-permissive in that particular environment. This is a hypothetical situation, however, and in reality, the epithelial cells that influenza infects are in the respiratory tract, and they are surrounded by a protease clara, which is being secreted into the local environment. And this protease clara processes the newly released virus particles and makes them infectious. So therefore, these cells can be considered susceptible and also permissive. So we have tropism. There's an additional factor that I haven't talked about, but it's useful to know, and that factor is accessibility. So a virus needs to physically access the cell type it's infecting. If if we have a cell type that is susceptible to infection by a particular virus and it's permissive to infection by a particular virus, that doesn't mean much at all if the virus can't physically access those cells. So if the virus can't physically infect those cells, it doesn't really matter if they're permissive, it doesn't really matter if they're susceptible, because there's going to be no infection. So when we're talking about this concept of tropism, we're talking about what cell types the virus can infect, and also it does infect. So we're talking about actual events that happen. So when we're talking about tropism, we're talking about a combination of susceptibility, permissivity, and accessibility. Again, which cell types can the virus physically access? So when we're thinking about what cell type any particular virus is tropic for, we're thinking about what cell type is susceptible, what cell type is permissive, and what cell type a virus has access to. So these are the three factors that impact tropism. Okay, so now we'll go on to talk about virulence. So yeah, this is a topic that, or a concept that is slightly distinct from pathogenesis. In some ways, it's actually quite distinct from pathogenesis. But either way, we need to know the distinction. On the surface, they can seem the same, but at level 6, we don't really want to be looking at things on the surface. We want to be getting into the details. So yeah, it's quite important that we understand what virulence is. You'll often see the two terms used interchangeably, so virulence and pathogenesis. However, in the literature you'll be reading, because you're level 6 students, you'll be reading peer-reviewed scientific reports, articles in journals and things like that. The terms won't be used interchangeably. The term virulence or the term pathogenesis will be selected when either is appropriate. So again, really important we understand the difference. When we're talking about virulence, we're talking about the degree of disease. When we're talking about pathogenicity, we're talking about the type of disease that's caused or the way disease is caused. So again, this subtle distinction, that's actually quite important. The main thing we focus on when we're talking about virulence is the presence of what we call virulence genes. And virulence genes are specific genes that encode factors that impact the ability of a microorganism, in this case viruses, to cause disease, essentially. So whether or not the virus has or doesn't have these particular genes determines the degree of disease it causes. So again, that's why they're called virulence factors. There's a slight distinction here to make between the root words for virus and virulence. So even though the root words might be the same, when we're talking about virulence, we're not necessarily just talking about viruses. So we are talking about just viruses here because this is a virology module, so we only care about viruses really. But elsewhere, you'll see the term virulence factor and virulence gene used in the context of bacteria and even potentially fungi and other microorganisms. That's because the word virulence doesn't specifically refer to viruses. it just refers to the ability of a microorganism to cause a particular degree of disease. So bacteria have virulence factors. Virulence factors aren't only found in viruses. Okay, so a little bit more information about virulence factors, which are encoded by virulence genes. So the virulence genes are the genes themselves. The virulence factors are what they encode. So we can generally split virulence genes up into these different categories, and it's pretty clear to see how a gene encoding a protein that belongs to each of these factors, or any of these factors, will impact the ability of a virus to cause disease. So the presence or absence of these genes will impact the degree of disease, the virulence. If a virus is lacking specific virulence genes, then it is less capable of causing disease, so less severe disease is experienced. So there's a lower degree of virulence. The four groups we have here are enhancing viral replication. So we have virulence genes that enhance viral replication. We have genes that counteract the immune response. And we have genes that have direct toxic effects on the host. And then we have genes that somehow enhance the spread of the virus, whether we're talking about cells within a host or between hosts. These genes enhance the spread. So genes belonging to each of these categories have the capacity to increase the ability of the virus to cause disease, so therefore have the capacity to increase the degree of disease caused. Another way of phrasing that is it's pretty clear for us to see how a virus that is deficient in a particular gene that plays one of these four roles, that has one of these has the ability to do one of these four things, a virus that is deficient in that gene would potentially be deficient in its ability to cause disease. So it would potentially cause a lesser degree of disease. Okay, so what we want to do now is look at some specific examples. At this point, we want to acknowledge that individual virulence genes can fulfill more than one of the roles we've identified on the slide here. So in this example, we have ICP34.5, and what this protein is doing is it's counteracting an immune response. The immune response is mediated by a factor called PKR. This is a cellular factor. When this PKR recognizes viral DNA, so a composition that is characteristic of viral DNA, double-stranded RNA. We know there should never be double-stranded RNA in a host cell, so if there is, it's indicative of a viral infection. So once PKR recognizes double-stranded RNA, it inactivates EIF2A, which is it's a cellular enzyme essentially. It's really important in translation. So when it's inactivated, we don't have translation. Cell essentially shuts down. So this is an immune mechanism to shut down an infected cell. ICP34.5 is a herpes virus protein that counteracts this. So it activates protein phosphatase 1A, which is a protein that essentially undoes what PKR has just done. So it dephosphorylates the protein that PKR has phosphorylated. Now, clearly this protein, this herpes virus protein, ICP34.5, is counteracting the immune response. It's directly counteracting this specific immune response. However, in doing so, it's enhancing viral replication because it's preventing the infected cell from shutting down. So it's enabling the herpes virus to continue replicating within the cell. So this virulence gene is fulfilling these two functions. We have another example here. We have NSP4. So NSP4 is a protein. It's produced by rotaviruses. And I think we've briefly talked about rotaviruses before. They're essentially causing gastroenteritis and diarrhea. So they're infecting the gastrointestinal tract and causing gastrointestinal symptoms, essentially. NSP4 is a protein, so a toxin produced by rotaviruses. It's an enterotoxin because of where it acts. it's secreted from infected cells essentially, and it has a specific impact on the local environment. So I've identified it as having a role in having a toxic effect on the host, and also enhancing virus spread either within the host or between hosts, so to a new host. The reason for this is NSP4 is directly implicated in the diarrhea that rotaviruses cause. So we can consider it a toxin, an enterotoxin, and this toxin directly causes diarrhea. Pretty clear, therefore, that this toxin has a direct toxic effect on the host. So we have role C in our virulence gene list fulfilled. We also have role D fulfilled because by inducing this diarrhea, NSP4 is mediating the release of huge amounts of virus particles into the environment in diarrhea. So it's essentially promoting the spread of the virus to new hosts by inducing diarrhea. Okay, so that's virulence. It's pretty clear how those virulence factors, well, hopefully clear how those virulence factors, whether they're present or absent, directly impact the degree of disease caused. So if we imagine viruses that we've talked about without their virulence factors, it's pretty clear to see how they wouldn't be able to cause the same degree of disease. You could also argue that, okay, well, they wouldn't cause the same type of disease, right? And the type of disease is related to pathogenesis. That's what pathogenesis is. Yeah, that's pretty much true. So that's why we say virulence and pathogenesis are overlapping concepts, but they are technically distinct. Okay, so what we want to do now is think about these concepts of direct and indirect pathogenesis. So we've established what pathogenesis is, we've established what virulence is, we've discussed some information around these concepts. What we need to do now is discuss specifically a distinction that we have within pathogenesis, where we say pathogenesis is direct or indirect. We've talked about this in quite a limited way, but to a degree in the medical microbiology module that's done at level 6. What I'm going to do now is give you specifically the information that is relevant to this module. Okay, so the key bit of information here is that we can classify pathogenesis as direct or indirect. So viruses, like other microorganisms, can cause disease in either a direct manner or an indirect manner. And if we break down the way in which different viruses cause disease, we can generally say, oh, this is direct disease or this is indirect disease. If we want to break down these definitions of direct or indirect, we can generally say that some microorganisms will, just directly cause disease. The action of the microorganism, by being there, by things it releases, by things it does, it directly causes disease. However, sometimes it's the either the immune response to the virus being there that causes a disease, or it's kind of and you can almost say an inadvertent effect of the virus being there. So it's not a part of the virus's life cycle. It's not something specific that the virus does. It's just an indirect consequence of it being present. So again, it's this distinction between direct pathogenesis and indirect pathogenesis. Generally, when we're talking about indirect pathogenesis, we're talking about the immune response. For other microorganisms, there are some kind of vaguer ideas of indirect pathogenesis. So, for example, things like ascariasis, where you'll get parasitic worms that accumulate in the lower GI tract, and they cause a physical blockage. That's kind of indirect pathogenesis, particularly if that blockage comes about when you treat someone with antiparasitics. So if you have a huge parasitic worm in an individual and then you treat them with antiparasitics and you kill that worm, and that dead worm can often just form a ball, like a physical blockage that blocks the GI tract. So it's difficult to consider that direct pathogenesis. That's really indirect pathogenesis. But those kind of situations are a bit outside of virology. So inside virology, again, they're outside of virology because I was talking about parasitic worms. We don't really have those issues and those grey areas in virology. In virology, when we're talking about indirect pathogenesis, we're really usually talking about the immune response to the virus's presence causing disease. The best example of direct pathogenesis, something we actually talked about a little bit earlier, it's this idea of lysis. So some viruses will directly cause lysis of cells they infect. We call these viruses cytopathic, cyto meaning cellular, pathic again from pathos. So they're causing the death of the cell. Cytolysis, the cytopathic viruses causing cytolysis, so causing lysis of the cells. Again, really clear example of direct pathogenesis. You don't need to know this diagram on the right, by the way. This is just if you want some more outside reading. This is from MIMS Medical Microbiology, and it's really good. There's a lot of information in the textbook around this figure. I don't really want to go into this figure in detail, but if you're struggling with the concept I'm talking about now, if you go to MIMS Medical Microbiology, you'll see this figure, and you'll see, I think it's about half a chapter talking through these concepts. Yes, so I'd encourage you to access that if you're struggling. But if not, then this should be pretty straightforward. Again, here we're just talking about the ability of the viruses to directly lyse cells. We talked about how for HIV, a part of HIV pathogenesis is the ability of the virus to lyse CD4 T cells. Poliovirus is another virus we use an example. So poliovirus replicates inside motor neurons, and it directly causes lysis of these motor neurons, which results in paralysis essentially. You're having muscular atrophy because you're losing these cells. Rotavirus as well. Rotavirus will result in lysis of enterocytes, so cells of the GI tract essentially, cells of the lower GI tracts of the intestine. Once these cells are lysed, it results in malabsorption. So that just means abnormal absorption, so you don't get the normal absorption levels, which means there's fluid loss because the water is, it's being retained within the lumen of the intestine, is not being absorbed by the cells that are lining the intestine. So ultimately this results in diarrhea. So again, a pretty clear example of how viruses are directly causing pathology through the lysis of cells. And again, just to bring it back to indirect pathogenesis, when we're talking about indirect pathogenesis, we're talking about pathology that is caused not by the direct action of the virus, but is instead caused by the immune response to infection. And again, we can see here on the right, we have kind of indications here of the indirect mechanisms that can take place, including endotoxins. This is really, really complicated, and it's beyond the scope of this lecture. So again, if you want to go to MIMS Medical Microbiology, these are talked through one by one. Just a little bit beyond the scope of this lecture. I don't really want to spend a whole bunch of time on this. We will spend time looking at some of these mechanisms in a later lecture when we talk about the immune response to infection. I just don't really want to get bogged down into it now. You'll see at the top of this diagram, we see as part of direct pathogenesis, there is, obviously we have cell lysis, which we've talked about, cytolysis, but we also have exotoxins. Now we talked earlier about how some viruses can release exotoxins. We talked about NSP4, which is a really good example of this. However, generally exotoxins are related to bacteria. We have a whole host of examples of different bacteria releasing exotoxins. So releasing exotoxins that directly cause tissue damage, that directly cause symptoms of disease. We have some examples for viruses, again, like NSP4 we were talking about, but usually when we think about exotoxins, we tend to think about bacteria. Definitely not absolutely true. Definitely some viruses that release exotoxins, but we generally just tend to think of them as being bacterial. You have endotoxins listed here as well. Then you'll see endotoxins feed straight into the immune response. So endotoxins are just factors that are present on a microorganism that induce an immune response. So that makes sense why, if we look at this diagram, we see endotoxins are linked to indirect pathogenesis, because a lot of indirect pathogenesis occurs when the immune system detects endotoxin. Again, a large number of endotoxins are found on bacteria. We tend to think of something called LPS, lipopolysaccharide, as the archetypal, as the main endotoxin. And again, LPS is found exclusively on bacteria. Okay. So finally, I have a few slides discussing oncogenic viruses. We're not going to go into this in any substantial level of detail because we'll get a little bit bogged down. But I just want to acknowledge the fact that some viruses are capable of causing disease in a way that seems to be outside of the normal mechanisms we've talked about. It's not really outside, like it all makes sense and it all obeys the principles. It just seems a little bit different. For example, based on all the principles we've talked about so far, it might seem unusual that viruses are capable of causing cancer, or it might be unclear how viruses are able to cause cancer, even though I'm sure you know that viruses are capable of causing cancer. So what I want to do is just approach that topic now. So we refer to viruses that cause cancer as oncogenic. We talk about the oncogenic viruses. We can also refer to them as tumorigenic viruses. Again, genic meaning the origin of something, onco meaning cancer, tumor meaning tumors. So whether we're saying oncogenic or tumorigenic, we're talking about viruses that induce cancer. Viruses do this by dysregulating what we call the cell cycle. So the cell cycle is the normal cycle through which cells progress to replicate. This is a really tightly controlled process. Obviously, it's not in the host's interest, it's not in our interest to have cells that are replicating in an uncontrolled manner, because that's what cancer is. Cancer is uncontrolled replication of cells. So therefore, the cellular replication cycle is really, really tightly controlled, and there's these really stringent control mechanisms that prevent uncontrolled proliferation from occurring. Now, what viruses that cause cancer do, so what these oncogenic viruses do is they cause dysregulation of this cell cycle. So when they infect a cell, they cause, by a variety of mechanisms, this cell cycle to start to occur in an uncontrolled manner. So it's no longer a tightly controlled, astringent process. It is an uncontrolled process that occurs over and over and over again, that results in uncontrolled proliferation, which ultimately results in tumor formation. Because again, that's what tumor formation is. It's the uncontrolled proliferation of cells into a physical mass. So even though on the surface it can seem somewhat unusual that viruses can cause cancer, that they can cause the cells to become cancerous, this specific process, by the way, is called transformation. So having a cell go from a healthy cell with controlled regulation to controlled replication to having uncontrolled replication, so this constant proliferation, we call this process transformation. So some viruses are capable of transforming cells. Okay, so again, whilst this process can seem unusual, whilst the idea that a virus infection in a cell can cause it to replicate and replicate and replicate, it really is just a mechanism of direct pathogenesis, right? The virus is coming into the cell, and by virtue of coming into the cell, it's causing the cell to proliferate in an uncontrolled way. Again, we tend to think of direct pathogenesis as involving cytolysis, so the death of cells, and in some cases, the release of exotoxins. However, when we're talking about cellular transformation in the context of oncogenic viruses, we are really talking about direct pathogenesis, just an unusual mechanism. I mean, a mechanism that doesn't involve cytolysis, it involves proliferation of the cell. It's also really important that we understand that cellular transformation is a rare event. It's a very rare event. So obviously cancers caused by viral infections occur, and they're widespread. You know, if we're thinking about cervical cancers caused by HPV, they're widespread. We have other cancers that are caused by HTLV and other viruses as well. These definitely occur. However, as specific events, they're fairly rare, shall we say. It's not commonplace. It's not every cell that's infected with HPV becomes transformed and that replicates in an uncontrolled way. So again, it's not a definite fact that a cell that is infected with HPV will become transformed and will form a tumor, but obviously it can happen, and it does happen. And when it does happen, it makes sense to think of it as an example of direct pathogenesis. Okay, well, thanks everyone. That's the end of the lecture for today. Again, hope you found it useful. If there was anything you didn't understand, as always, please do go back through the slides, have a listen again, access the resources that are there to support you, so the textbooks, maybe read some research papers if you think that would be useful. I think that is a good approach sometimes, particularly reviews. Literature reviews can be really helpful and often quite accessible. We also have the tutorial on Wednesday, so I'd always encourage you to come to the tutorial, and if you have any questions, come prepared with those questions, because I'll always be happy to answer. Okay, thanks very much. Take care.